Hello everyone, it is my pleasure to introduce to you an extraordinary aspects of the animal kingdom, homeostasis and waste removal. These two processes plays a critical role in helping animals survive and thrive in their environments. This report explores the crucial mechanisms of homeostasis and waste removal in animals, highlighting their significance in ensuring the survival and optimal functioning of organisms. It examines the various strategies employed by different animal species to maintain internal balance and eliminate metabolic waste. Animals have evolved a remarkable ability to maintain stable internal conditions, allowing them to survive in diverse environments. So let's jump on the definition and importance of homeostasis. Homeostasis is the maintenance of the internal conditions in response to external changes. It is crucial for an organism's survival and ensures optimal functioning. Homeostasis is not static nor unvarying. It is a dynamic process that can change internal conditions as required to survive external changes or challenges. If you ask me, why is it dynamic? Because it is constantly adjusting to the changes that the organism's body systems encounter. So, mechanisms of homeostasis. Adjustment of physiological systems within the body is called homeostatic regulation, which includes the three parts of mechanisms. First is the receptor. It receives information that something in the environment is changing. The control center or integration center receives and processes information from the receptor. Then the last one is effector. Responds to the commands of the control center by either opposing or enhancing the stimulus. The process of homeostasis can be illustrated through the examples of body temperature regulation. First, example of the receptor. It is specialized temperature-sensitive receptors called thermoreceptors detect in changes body temperature. They are located throughout the body, particularly in the skin and central nervous system. Here, example control center is the hypothalamus or the region in the brain. Acts as the control center for temperature regulation. It compares the information received from the thermoreceptors to the set point, which is typically around 37 degrees Celsius or 98.6 degrees Fahrenheit. The last one is effector. If the temperature deviates from the set point, the control center sends signals to effector to bring about the necessary changes. If the body temperature is too high, examples when you are exercising or exposure to hot weather, the control center sends signals to the sweat glands and produce sweat for evaporative cooling. Temperature regulation. Animals employ various strategies such as thermoregulation to regulate their body temperature in both warm and cold environments. Parang adapting siya in which environment you are in. Osmoregulation. This mechanism maintains the balance of water and electrolytes, preventing dehydration or overhydration. pH regulation. Animals have buffer systems that control the acidity or alkalinity of bodily fluids, maintaining an optimal pH for metabolic processes. And the last mechanism is nutrient regulation. Animals regulate nutrient intake and metabolism to ensure a balance of essential macronutrients and micronutrients.
There are three types of organism in terms of their extraction and handling waste materials. The first type of organism is a monotelic organism. It is excrete their waste in the form of soluble ammonia. And second type of organism is oriotelic organism. It is produce urea as waste material. And lastly, type of organism is Oricotelic organism. It is generate uric acid. Among these three forms of waste, ammonia is the most soluble in water, and uric acid is the most insoluble. Aquatic invertebrates and fishes are ammonitic and secrete large amounts of ammonium ions through their gills. Animals such as mammals, sharks, turtles, and saltwater fishes are eutelic, while birds, insects, snails, and many reptiles are oricotelic. These are the differences between ammonitic, eutelic, and oricotelic organisms. There are four steps in production of urine, namely filtration, selective absorption, selective secretion, and extraction. So the first step in production of urine is filtration. So filtration is a process that occurs in the kidneys where the blood is filtered to remove excess water, salts, and other waste product from the body. This filtered blood then enters the renal tubules where it is further proceed to form urine. So the next step is selective absorption. This is the process in which the kidneys filter out certain substances from the blood into the renal tubules. During this process, glucose, amino acids, vitamins, minerals, and other molecules are absorbed back into the bloodstream, while urea, salt, and other waste products remain in the tubules and are eventually excreted as urine. The next step is selective secretion. This is the process of reabsorption and secretion of specific molecules from the glomerular filtrate by the nephron to produce urine. This process takes place in the renal tubular cells of the nephron. During selective secretion, substances such as sodium, potassium, calcium, chloride, and bicarbonate are actively reabsorbed from the glomerular filtrate. At the same time, other substances such as urea, creatinine, and uric acid are actively secreted into the filtrate. This process of selective secretion helps to maintain the balance of electrolytes and other substances in the blood. And the final step in production of urine is extraction. It involves the movement of urine from the kidneys through the ureters into the urinary bladder and finally out of the body through the urethra. Generally, the chylamic fluid, blood, or hemolymph is collected and filtered by a semi-permeable membrane called transport epithelium that prevents the passage of large molecules while small solutes and water are allowed to traverse. The resulting filtrate undergoes selective absorption, wherein molecules are returned into the body depending on the current requirements. Toxins, excess salts, and other waste materials are then pumped out. These solutes then attract water for dilution. The combination of waste and water is then extracted in the form of urine. Organisms have developed different types of extractory systems, namely protonephridia, that is present in some annelids, larval stages of mollusks and flatworms. Their extractory system is characterized by a network in tubules. Selective absorption and secretion occur in the tubules and moves outward into the environment through the nephridopores. Most annelids are metapradians. A pair of metapradia is present in each segment of the body of the annelids and is surrounded by capillary network. The fluids are pushed from the capillary blood into the coulombic space, then enter the excretory tubules via the nephrostome. Insects and arthropods contain malpighian tubules. These tubules outpacket from the digestive tract with the closed tips immersed in the hemolymph. Solutes are actively secreted from the hemolymph towards the tubules, and water molecules follow them via osmosis. Vertebrates have a kidneys with a main filtering unit called nephrons. Each nephron contains structures, namely the Bowman's capsule that encapsulates capillaries called the glomerulus. Blood pressure forces the waste materials and other solutes out of the capillaries. These substances are collected by the transfer epithelium of the nephrons and are extracted out of the body. The nephrons also have the law of Henle that facilitates the regulation of electrolyte concentration and water balance in the body through selective resorption and secretion. So mao ni ang Malpighian tubules kanang naa sa baba ng hangouth na mula giring-giring.
Here is the picture that defines Bowman's capsule, glomerulus, and nephron.
Naasad ang lobe Henle para mag-facilitate sa regulation of electrolyte para mag-concentrate og ang water balance ng body ma-selected na reception and secretion. That's all. Thank you.